Welcome to the Ask the Expert. We're here with Dr. Andres Garcia. He's coming to us from Georgia Tech. And the title of this talk is Biomaterials for Transplantation of Insulin-Producing Cells. Dr. Garcia is the Executive Director of the Parker H. Pettit Institute for Bioengineering Bioscience, the Pettit Director's Chair. He also has a Pettit Director's Chair in Bioengineering and Bioscience, and he's also a Regents Professor, George W. Woodruff School of Mechanical Engineering at Georgia Institute of Technology. So his research program integrates innovative engineering, material science, and cell biology concepts and technologies to create cell-instructive biomaterials for regenerative medicine and generate new knowledge in mechanobiology. This cross-disciplinary effort, which we laud, has resulted in new biomaterial platforms that elicit targeted cellular responses and tissue repair in various biomedical applications. Innovative technologies to study and exploit cell adhesive interactions and new mechanistic insights into the interplay of mechanics and cell biology. And in addition, his research has generated intellectual property and license agreements with startup and multinational companies. He's the co-founder of 3 startup companies: Select Cell, that's C-E-L-L-E-C-T, C-E-L-L, Core Ami Therapeutics, which is C-O-R-A-M-I, and I Tolerance. He's received several distinctions, including an NSF Career Award, Young Investor Award for the Society of Biomaterials, Georgia Tech's Outstanding Interdisciplinary Activities Award, and the Klenzel Award for Basic Science from the Society of Biomaterials. And finally, let's see what else, a lot more. The International Award for the European Society for Biomaterials and Georgia Tech's Class of 1934 Distinguished Professor Award. He's the elected Fellow of Biomaterial Science and Engineering by the International Union of Societies of Biomaterial Science and Engineering, a fellow at American Association for Advancement of Science, and fellow of the American Society of Mechanical Engineers. He served as the president for Society for Biomaterials in 2018-2019, and he's an elected member of the National Academy of Engineering, National Academy of Medicine, and National Academy of Inventors. Wow, he's very, very busy and with a very deep CV. Welcome. I'm really interested to talk to you today about, your work in biomaterials for islet transplantation, and you want to kind of set the stage and, give us a little bit of a background on what you've been up to.
Great. Thank you, Monica. It's really a pleasure to be here with you and your audience. And I'm excited about sharing with you some recent data and results. And as you mentioned, my lab focuses on engineering biomaterials for islet and cell transplantation, including for the treatment of type 1 diabetes. Yes. So as many of us know, islet transplantation is really, to date, the only effective potential cure for type 1 diabetes. And this has been shown to be safe and effective for type 1 diabetes patients, but the clinical trial involved patients that had hypoglycemia unawareness and severe hypoglycemic events. While this is very promising, it's not a durable cure, and most of these patients go back onto full insulin. So, a lot of excitement in the field, especially recently with the Vertex data, with the human stem cell derived insulin-producing cells. So I think, the cell therapy to treat type 1 diabetes is really at a cost of really advancing and really, improving the life of the patients. Yeah. So as summarized here, there are some significant limitations to have islet and stem cell based therapies as a broad cure for type 1 diabetes. The problem that we have been working on, and I'll talk about, deals with the immune rejection to this allogeneic, in the case of islets, right? Because they're obtained from genetically dissimilar cadaveric donors that provide a mismatch, and the need and complications associated with this heavy immunosuppressive drug. So recipients of either islets or human-derived insulin-producing cells must remain in long-term chronic immunosuppression And those drugs are toxic not only to the recipient, but also for the graft. So
yeah, and it's also very difficult to get, you know, the younger patients involved in that kind of treatment. And many of the patients who do get type 1 diabetes get that disease at a young age.
yes, and you know, balancing the risks to benefit is really a very difficult thing. So, you know, the goal of really reducing or potentially eliminating these immunosuppressive drugs is really an important topic in the field.
Absolutely.
So there are many groups that are working on this, and you know, there's been long-running strategies, for example, using biomaterials to encapsulate islets. And unfortunately, today, those approaches really have not proven success in either large animals or humans. So our approach has been different in that we are focused on engineering immune tolerance to the graft, right? So the idea or the concept is, can we induce the recipient to accept the graft without the need of immunosuppression?
The
So
holy
to do
grail,
that,
really.
yeah, no, it's been, and again, this is important not only for you know, cell therapy in diabetes, but it's also important in many other applications. So our strategy has centered on delivering a very potent immunomodulatory protein, Fas ligand, as a therapeutic target, right? And Fas ligand is a key modulator of peripheral tolerance in mammals. And in the context of cell transplantation, we know that activated T cells express Fas receptor and become sensitive to Fas ligand apoptosis. And again, this is the major mechanism by which we create localized peripheral tolerance. So earlier, proof of principle studies demonstrated that indeed Fas ligand induced apoptosis to block T effector cells from killing islet grafts was a promising strategy. And much of this work was done using gene therapy. And while exciting, you know, these approaches face significant technical and translational limitations associated with off-target effects. And Monica, as you indicated, this is primarily a young population. So, you know, there's concerns there.
Yeah. I mean, you have to be it has to be specific and scalable, right? That's the name of the game.
Correct. So, you know, and other work demonstrated that the format of the presentation of the Fas ligand, so when using the protein, the format of its presentation was critical to function. And for Fas ligand to work properly, it had to be immobilized on the surface of a presenting cell. So that was important for us from a biomaterials perspective because it suggested a mechanism for the delivery. Mm-hmm. Now, the target that we use of Fas ligand is a recombinant chimeric protein of Fas ligand that was developed by our collaborators, Esma Yoko and Haval Sherwan from the University of Missouri. And this protein contains, and the schematic here shows it, it has multiple copies of the extracellular domain of Fas ligand fused to the streptavidin core protein. Okay, and in a very elegant study, they had demonstrated that if they chemically modified islets to present this protein, they could induce immune acceptance. Mm-hmm. So the challenge there is that you're chemically modifying the islets, and again, this poses challenges, as you mentioned, when you're trying to develop something that's translatable and scalable, right?
Right. And so can you just dig into that a little bit? So you guys are modifying all cells that are, you know, being transplanted as islets. So or just beta cells?
No, so the strategy that they had previously shown was modifying the islet. So it would be all of the cells in or a significant number of the cells. Mm-hmm. But this prior knowledge really served a foundation for the strategy that we wanted to pursue. And that was, you know, let's engineer a biomaterial for the controlled presentation of the Fas ligand without having to chemically modify the cells, right?
Yeah, that better.
We agree, right? So the idea was to generate hydrogel particles, and which are called microgels. These are hydrogel spheres, and we make them by microfluidic polymerization. And you can see how this droplet generates the particles of control size that become cross-link and become hydrogels. And this movie just shows that by changing the flow rate, we can change the size of the resulting particle. The important thing is that this method really provides a well-controlled approach to engineer fully synthetic hydrogel particles, right? So we have complete control over the chemical and mechanical compositions of this biomaterial, and we have it with very high precision.
Okay? Yeah, that's great.
So the idea was to make a particle that was functionalized with biotin, which is a small vitamin, and use that biotin to capture and present the streptavidin Fas ligand, because streptavidin binds biotin with high affinity, right? So that was the concept. And in fact, that works very well. You can see here microgels labeled in red, ones that have biotin and ones that do not have biotin. And only those that present biotin can capture and present streptavidin labeled proteins.
Nice.
Right? So similarly, we can engineer microgels that have a controlled density of the streptavidin Fas ligand on the surface simply by varying the coating concentration. And importantly, we show that engineering this biomaterial maintain the bioactivity of that Fas ligand. So we test that in lab in the culture by looking at apoptosis on a target cell, and you can clearly see that we have dose-dependent increase in apoptosis only when we display Fas ligand using the biotin from the biomaterial. So these studies show that we can engineer a material with controlled density of Fas ligand that retains its biological activity. So then, the question is, does this work in the setting of islet transplantation?
Yeah.
So what I'm going to summarize here is really the key study that we did first in mice and then on a large animal model. So first, the murine study. So this is an allogeneic transplant model, right? So we isolate islets from one strain of mice that is immune differently than the recipient mice, which are diabetic, right? So in this model, the islets should reject very quickly. And then we assess whether co-delivering the microgel particles presenting the Fas ligand had any positive effect.
And how are you delivering these? Just, by injection to the local region?
So these are delivered on the paper. On the study, we examined 2 transplant sites. One is under the kidney capsule, and the other one was in the epidermal fat pad, which is analogous to the omentum in the larger animals.
Right.
Monica, one important thing here that I need to stress is the islets were not encapsulated in the biomaterial and were not manipulated. We simply mix the islets with the biomaterial at the time of transplantation. So, from a translational standpoint, this is very attractive because it could be an off-the-shelf application.
Oh, yeah.
Right?
And yeah, so then how long did it last?
Right. So what we're looking here is survival of the graft as measured by the ability to reverse diabetes in this chemically induced mice as a function of time, right? So if we look first at the control, so the black circles are islets co-transplanted with control microgels that did not have the protein, the Fas ligand. And you can see that islets in that group rejected very quickly. Within 25 days, all of the mice rejected the graft, showing that this is a stringent model of allo transplantation. The blue triangles are islets co-transplanted with the fast ligand microgels. And you can see that we have an increase in the lifetime of the graft. And importantly, about 25% of those recipients have long-term graft acceptance for up to 200 days that could function properly. Again, this is without any chronic immunosuppression, so that's a significant advance.
And you added rapamycin here, right?
Yeah, and the reason for that is our colleagues had demonstrated that if you combine the fast ligand with a brief dose of rapamycin, 15 days post-transplant, we could boost the immunomodulatory efficiency or effect of the fast ligand. And that's what's shown in the red curve. And the data is remarkable, right? Over 93% of those recipients have long-term graft acceptance and function without any chronic immunosuppression. And by all metrics, these mice were cured and restored to normal glycemia. So for example, if we look at graft function via a glucose tolerance test, so what's done here is at sacrifice, we inject a glucose bolus, and then we see how quickly it's cleared from the blood, right? That's a measure of graft function. You can see here curves for the fast ligand microgels with the rapamycin recipients at 200 days compared to naive healthy mice. And you see that for both cases, we can easily clear the glucose challenge, right, in the normal kinetics, whereas the mice that had rejected grafts have very poor glucose disposal. And again, when we look for staining at the graft site, the ones that had the fast ligand microgels, we could find insulin positive structures that reflect the transplanted islets in close opposition to where the microgels were co-transplanted with the fast ligand. Whereas the controls that did not have the fast ligand, there was no insulin remaining at the take.
So the microgel stays, basically with the islet then.
Correct.
It doesn't diffuse and, impact other organs.
No, exactly. The microgels are about the size of the islet. So they're about 150 to 200 microns. So they stay locally. So, this is, again, very promising and exciting results. We did a fair amount of immune analyses to identify the mechanism by which this was working. And interestingly, we found that the actual fast ligand protein only remains there for a very brief time. So for about 4 to 7 days. By 2 weeks, all of the protein has cleared. But what we see is that in that brief induction window, the biomaterial induces a population of T regulatory cells, or Tregs, and those are the cells that establish an immune acceptance circuit and maintain graft acceptance. And if you go to the publication, you see that if you deplete or you ablate those cells later, the graft rejects, right? So the biomaterial induces this very potent population of T regulatory cells, and it is those cells that maintain the graft acceptance.
What, what flavor Tregs are those?
What do you mean?
Well, can you elaborate, like what Tregs are called over?
So these are T regulatory cells that induce and maintain this immune acceptance. These are your CD25 positive, FOXP3 positive Tregs.
Right. Great. This looks very promising.
So, we then, and again, we were very fortunate to be funded by the JDRF. We then were able to test this approach in diabetic monkeys, right? And this is a very challenging preclinical model in non-human primates. And we basically did the same thing that we did in the mouse study. So we co-transplanted allogeneic islets, again, mixed together with the fast ligand microgels. in a diabetic non-human primate model without chronic immunosuppression. Okay, and this data was recently published. So if we look at the metabolic outcomes, so on this graph, I'm plotting the average blood glucose levels, which correspond to the blue line. Anything above the hash line is hyperglycemic, right? So we want to be below the hash line. And the orange bars refer to the external insulin dose that's given, because in these recipients, we did marginal grafts, right? So you don't expect that one donor to one recipient will completely return normal glycemia. But the results are here. These were 4 treated animals. We see that prior to transplantation, there was very poor blood glucose control. These animals also needed a high dose of insulin to control their blood glucose levels. But post-transplantation, we quickly see reversal to normal glycemia. We have excellent blood glucose control for over 6 months. Also, these recipients had reduced insulin dosage to about 10 times the original pre-transplant dose. And in fact, one of the recipients by the end of the study was completely off exogenous insulin. Again, this is over 6 months without the use of any chronic immunosuppression. The RAPA regimen was modified a little bit to go up to 90 days, but certainly after removal of RAPA, we had very good blood glucose levels. To demonstrate that this reversal was indeed due to the graft, we explanted the graft in the recipients, and that demonstrated complete and quick reversal to hyperglycemia, right? So that's an important study because it demonstrates that the reversal of the diabetes is due to the graft transplantation.
Yeah, that's very key.
So those were the 4 treated animals. We also had 3 control monkeys that receive islets, microgels without fast ligand, and the same rapamycin regimen. And those recipients very early demonstrated signs of graft rejection and very poor glucose control, as you would expect, in this allotransplant model. So summarizing the graft survival, we see that we co-transplant islets with the fast ligand microgel. We have indefinite graft survival over the duration of the study, whereas the controls reject very quickly.
Right.
And then to share, and these are really remarkable data, so we're very excited by them. And then a little bit more information just to round up the study. Again, for the fast ligand treated recipients, we see normal insulin and C-peptide levels as shown here, right? So this is the naive levels, insulin on the left axis, C-peptide on the right axis, and we have measurements for both fasting and then stimulating. And importantly, you see that this is after they're made diabetic, 3 months and 6 months after transplantation. We normalize the insulin and the C-peptide to what we have to the naive state. And importantly, we see that there's actually glucose responsive C-peptide production, right? So when you go from the fasting to the stimulated one after they eat, you actually see an increase in both C-peptide and insulin, demonstrating metabolic responses to that. We also did a glucose tolerance test, and we show excellent disposal kinetics, similar to a healthy recipients. Again, because we had seen that Tregs were necessary in the rodent study, we saw that at the graft site, so locally, we saw an increased number of FOXP3 positive cells, which are, that's a marker for Tregs. So we see an enrichment of Tregs in the treated recipients, which is consistent with the mechanism of action that we saw in the murine study.
Yeah.
And then importantly, from a safety standpoint, we saw no changes in T cell systemic frequencies or responses to donor or third-party antigens, which suggests immune acceptance And, in initial studies, we saw normal liver and kidney metabolic function. The controls, as expected, had undetectable insulin and C-peptides, right? These were really not doing well. They had very poor glucose disposal. They had increased antibodies against the donor antigens and T-cell responses to the antigens, demonstrating that they do elicit a response to that transplant. So, very excited by the results on the non-human primates. There are very few studies that demonstrate such promising results in this very challenging, stringent, large animal model. And, this technology has been licensed by a startup that we co-founded called iTolerance, that is working to bring this to a clinical trial. So we have completed our pre-IND with the FDA. We know what needs to be included in the full IND, and we're working towards that. We are expecting to submit our full IND in the first half of 2023, and then to start the first in-human study in the second half of 2023.
That's very exciting. What is the, can you just comment a little bit on how this work might have the possibility to dovetail with the work being done at both the ViaCyte and Vertex?
Yeah, certainly. Thank you for the question, Monica. That's something that's very important to us. So the foundational studies that we have done are with cadaveric islets, as that already has clinical approval. But we're very keen to move this into human stem cell derived insulin producing cells, as this is really a more viable, renewable cell source. So we have a couple of programs within both the academic labs as well as the company to really focus on transplanting those stem cell derived beta cells. We expect that the technology will work just as well. As I pointed out, the technology is sort of agnostic to the cell source because we don't have to encapsulate the cells. We don't have to manipulate the cells in any way to present the protein. We simply co-mix them at the time of transplantation, right? And we have some data that suggests that that's fully compatible with the stem cell. So we don't anticipate any problems in moving this into the stem cells. And as you know, the clinical trials that have shown promising results with the stem cells, those patients are still in full immunosuppression.
Yes.
So we think that, our approach provides really an alternative route to the use of immunosuppression to induce immune acceptance.
Yeah, no, it's a very interesting approach for sure. And, with this implant, have you thought about delivery in a human setting? Like just at, the omentum again is a very popular site due to its, rich vascularization.
Correct. Yeah. So that's, the non-human primate study that were transplanted at the omentum. We had such positive results at that site that currently the plan in the human trial is to go to the omentum because, as you pointed out, because of the rich vascular supply. We do think, again, that this technology, and we're exploring other transplant sites that may have different advantages at the omentum, notably accessibility. So again, an advantage of this technology is that it's applicable to other sites because simply we're just putting biomaterial particles mixed in with the graft. Yeah.
And so I guess ultimately you could imagine this could be an ejection into the abdomen, if it all works and, is all optimized, an
Correct.
ejection into the omentum, into the abdomen. And then maybe it would last for you've got 6 months here. And then even if you had to do another injection twice a year, that's an incredible opportunity there.
Yeah, we do expect that this is going to last longer than 6 months. The constraints that we had on this particular study in the monkeys was that Due to COVID, there were some institutional restrictions that didn't allow us to continue taking the study. You know, the animals, when we would sacrifice them, they look great. You know, there were no signs that there were any degeneration in the graft performance. So we do expect to provide enhancements over longer periods of time. The question remains how long. But as you point out, you know, again, this strategy should be compatible with having to redose with more cells if necessary.
Yeah, which is, you know, fine. I mean, as it would still be ultimately an incredible, you know, incredibly a better situation than, you know, we have currently for type 1 diabetes. I guess I would also just ask, in terms of the, you know, in humans, you've got memory cells that are, you know, hanging out at certain sites, sort of unnamed sites, you know, wherever they are locally in the pancreatic lymph nodes, or maybe even in the appendix, or wherever they're hanging out. So, you know, do you feel that, you know, this whole, you know, the FOXP3 cells are going to be enough to handle it if those memory cells get reactivated?
Yeah, I mean, that's a great question. You know, also, and I will point out that remember the non-human primate model that we use here was a chemically induced model, right? It doesn't have autoimmunity, whereas in the human condition, you do expect some, you know, there's autoimmunity. You know, Monica, those are all questions that we're keen on looking at. I think they only can be addressed when we move into the clinical trial, and we will be looking into that. Based on our work before with the fast ligand and the induction of immune acceptance via Tregs, you know, we're excited, we're optimistic that this will work also as well in the clinic. But certainly we'll, you know, we will only know by doing the trial.
Right. I just wanted to invite some of our other attendees here to ask questions. We have a couple people sitting here that actually work in the space. So if they would like to unmute themselves, you know, happy to have some questions. I see Amir is here. Dr. Amir.
Yeah. Hi. Sorry. Can you hear me okay?
Yes.
Yeah. Thanks very much, Professor Garcia. I really enjoyed your presentation. I'm sorry, I'm not sure is my camera on there, but
yes, it is.
Yeah,
it is. Okay, perfect. Okay, great. You don't need to, but thanks very much. So my name is Dr. Emer Dolan. I'm based in Galway in Ireland. So it's great that I could join this call this evening to listen to your presentation. So I have 3 questions, if you don't mind. So my first one is just around the delivery. So you had, or Monica had postulated that potentially you could have some kind of an injection, you know, every year or so. Is the formulation viscous, or is it quite easy to deliver through a needle?
No, you can deliver through a needle because it's basically particles, right? So you're just injecting the particles in a suspension. So certainly, you know, depending on the size of the particle and the concentration, you could get something that's a little heavier. But just so you know, these ones for these studies, they're basically liquid quality, so not viscous at all.
So they wouldn't be affected by some kind of shear force.
No, we don't expect that that's going to be a problem.
Cool.
That's super. And then in the explant, so you had mentioned that you explanted them in the non-human primate study. How difficult was it to explant them, or how did you find the graft?
They were in the omentum, so these were going to be, it was a takedown. So getting the transplant area where we had put in the islets, we just took out the whole section of that omentum. And then we had to go through the sections to find the islets and the microgels. That's what was a little bit more challenging.
Okay, so it wasn't possible to find, like visually pick out the graft and the omentum? You just
we took the explant out and did the section in there. We did not. That was the design of the study, was to do it that way.
Super. Okay, and then my last question is around, and I apologize, I missed the first few minutes of your talk. But are the microgels eventually resorbed, or do they just stay put? Great
question. So the formulation that we use in this study is a non-degradable formulation. To give you a little bit more background, these are polyethylene glycol microgels that are about 95% water, so only 5% is the polymer. We designed them to be non-degradable. We do have formulations now that are degradable either by proteases or hydrolytic. So those are always an option if we really want to get complete resorption of the biomaterial.
Okay, that's great. Thank you very much.
Thank you.
And yeah, again, I really enjoyed your talk.
Thanks.
Yeah, that's fantastic. And is there, anyone from Syraxis here that would like to weigh in on this or ask a question?
Oh, hi, Andres. It was a good talk. I did have a question, but it was about the resorption of the PEG microgels, and you just answered it.
Okay.
However, let me. There's one thing I noticed on your slide. It had said that the range in diameter of the gels was between 20 and 400 microns, and I think as you were speaking through it, you limited that range to 200 microns.
Yeah, sorry. So yes, that's the range that we can make the particles with this technology. But we can have very tight control. So most of these are below 5% of the size that we target. So in the non-human primate, we picked a much narrower range of about 180 to 200 microns.
Okay, terrific.
So the 20 to the 400 is what we can make by microfluidic polymerization, but that's not the ones we transplanted. What we transplanted was a very narrow range.
Terrific. Well, thanks a lot. I appreciate it.
Yeah.
Thank you again. I mean, this is great. I really enjoyed, hearing from scientists who work in academia and industry in the same space, and hopefully, maybe some offline collaborations will develop as a result of this. But thanks again, Andres. This was a phenomenal talk, really pushing the envelope in this realm, and we can't wait to see really what you do next. So I hope you have a great rest of the day, and we'll talk again soon.
Thank you so much.